a second. Whoops. This meeting is being recorded. Yes. Continue.
Oh, then I'm leaving.
Please don't.
What would happen if I didn't check got it? Yeah. Yeah, that's fine.
All right. Hold on a second. All right. So it is my pleasure to introduce today's seminar speaker, Dr. Paul Gulig. He's a professor at the University of Florida College of Medicine and in the Department of Molecular Genetics and Microbiology. His current research interests are in drug discovery for Pseudomonas aeruginosa. And he said when he's not a professor, he's an outdoor kind of guy, biking, sailing, swimming, triathlons. And we were just talking about college sports. So he's been in Gainesville for 33 years. So he's a Gator fan and he's wearing proudly his Gator shirts. His research has focused on bacterial pathogens, and he started as a graduate student at the University of Texas Southwestern Medical School in Dallas. And he examined the immune responses to outer membrane antigens of Haemophilus influenza type B, which at the time was a leading cause of pediatric meningitis. And then he did his postdoctoral studies at Washington University in St. Louis and focused on the molecular pathogenesis of Salmonella enterica, Staphylococcus typhimurium. And so he used a variety of genetic manipulations and mouse and cell culture models to look at the role of the virulence plasmid in pathogenesis. And then in 1988, he joined the faculty of the University of Florida, and he continued his molecular pathogenesis studies on Salmonella. And then he changed to Vibrio, which is a pathogen in shellfish. And he studied a number of different bacteria, but right now his favorite is Pseudomonas aeruginosa. And then on top of that, he has a lot of non-research activities. He loves to teach, and he is also involved in administration. And I actually know Paul through biosafety stuff because we work together occasionally for Advara IBC, and he always does a really good job when he's presenting protocols, as exciting as you can make them on the IBC protocols. And so I thought it'd be really fun to have him here as a seminar speaker. So I'm looking forward to his talk, which is entitled The Winding Road of Development of Drugs that Inhibit Beta-lactamase and Type 3 Secretion in Pseudomonas aeruginosa. So Paul, all yours.
Okay, thanks, Chris. Let me move all of these little Zoom things out of the way. Yeah, okay, so as you can tell from Chris's introduction, my whole career has been molecular pathogenesis of bacteria. And I took over this project from my colleague, Shobong Jin, when he took a leave of absence. So that's why he's asterisked here. And the person who's done almost all of the work that I'm going to talk about is Young Kim, who's a postdoc in the lab. My current collaborator is Tamer Lange. He's in the College of Pharmacy, and you'll see his involvement all from the very earliest. This project is done in collaboration with Scripps, Florida. You know, their main campus is out in San Diego, and they have a satellite campus down in Jupiter, Florida, and now there's talk of them actually joining the University of Florida in a not a collaborative way, but becoming part of us. That's kind of exciting. And there are 3 faculty members down there: Tim Spicer, Tom Bannister, and Louis Scampavia, who have been instrumental. And then, of course, they have their own postdocs and other people. So this has been a really interesting collaboration, for me. So let's talk about Pseudomonas. So it's a gram-negative rod. It's found in the soil and water. So it's really an environmental opportunistic pathogen. It is the leading cause of death in people with cystic fibrosis. It basically infects their lungs and it never goes away, and eventually it kills them. If you get a burn wound, it's a great pathogen for that. Severely damaged skin, very, very big problem in the ICUs, the burn wounds. And I am going to see if I can get a pointer that is a, no, that's just a plain mouse. I don't want to draw. All right, never mind. Can you see the mouse okay?
Yeah, I
Eric?
can see it.
Okay, all right. Yeah, I know there's some way of getting a laser, but my experience is that ends up interfering with advancing the slides. Anyway, so people who are severely immunocompromised, neutropenic in particular, they're susceptible. My wife and I swim a lot. She's had some recurrent swimmer's ear infection, probably Pseudomonas. She didn't get it from me, from the lab. Hot tub folliculitis, and then eye infections. People who wear contacts, and I don't. These are reading glasses that I'm wearing. But some of the contact lenses have been designed, I think, to stay in for like days, if not a week or more. And the problem is, if you get some bugs behind that, they can cause some pretty nasty degenerative eye infections. And again, sepsis, systemic infection. So it's a big problem. Opportunistic infection, we encounter it all the time because it is in the soil and water. And it's the reason that you can't take flowers into the ICUs in the hospital. So it is notoriously antibiotic resistant. Its outer membrane makes it somewhere between 12 and 100-fold less. It's less permeable than E. coli, which has, I guess we'll say, more promiscuous porins. And it's notorious for having resistance plasmids that encode a whole plethora of antibiotic resistance mechanisms. And it, along with some other bugs, has these MEX OPR, a whole family of transport systems that, in a way that I find it difficult to understand, can transport out of the cell just wide varieties of unrelated antibiotics. So this bug is a real serious problem, and especially when you think about it causing infections in compromised people whose defenses can't fight the infection. The fact that it's resistant to antibiotics is a big problem. The one I'm going to talk about today is an AMP-C beta-lactamase. Now, most of us think about beta-lactamase as being on plasmids. A lot of the plasmids we use in the lab have the BLA gene in it. But this is a chromosomally encoded beta-lactamase. And what's important here is that its expression is induced by a regulator called AMP-R. And as you'll see later in the seminar, that's really a misnomer. I mean, it was discovered because it induces AMP-C, but it in fact has a much broader role. And so there's a periplasmic peptidoglycan fragment with this name here. It's a fragment of peptidoglycan. It binds to AMP-R, and AMP-R induces AMP-C. And so if you treat Pseudomonas with a beta-lactam and it starts interfering with peptidoglycan biosynthesis, this fragment is made, and that's kind of like a danger signal to the Pseudomonas. It gets transported from the periplasm into the cytoplasm by this AMP-G transporter protein. And then of course, the beta-lactamase is made and it inactivates the beta-lactam antibiotic. And that's kind of shown here. Here's the peptidoglycan fragment. Here's AMP-G. It gets processed by another enzyme, but then it works with AMP-R, turns on AMP-C, which makes the beta-lactamase, which degrades the beta-lactam. So the hypothesis for this whole project, which was started long before I got involved, if that peptidoglycan fragment is not transported by AMP-G, then AMP-C will not be produced and the beta-lactams will be restored in their usefulness. And so, if you're familiar with antibiotics and bacteria, there's a kind of a similar story with clavulanate or clavulanic acid. It though is a chemical that directly inhibits the beta-lactamase. And in fact, clavulanate is mixed with amoxicillin, and that's a very broadly prescribed antibiotic called augmentin. So the clavulanate will inhibit some of the beta-lactamases and allow the amoxicillin to do its job. And that actually works, but clavulanate does not work on AMP-C. So the endogenous chromosomal AMP-C doesn't care about clavulanate. So can we inhibit the transport of the inducer molecule by inhibiting AMP-G, and therefore it will not work with AMP-R, AMP-C will not be made, and the beta-lactamase will not be made, and the beta-lactams will be able to do their job. That was the hypothesis. So we go all the way back to 2010, and here are my two colleagues, Tamer Langehi and Chowon Jin. And again, Tamer was really the brains behind the initiation of this project. And he basically said, what's the role of AMP-G and can we possibly manipulate it? Okay, so they took the very commonly kind of the E. coli K12 strain of Pseudomonas PA01. And it turns out that cefoxitin, which is a beta-lactam, it induces the AMP-C beta-lactamase, kind of as I showed you. So here we're measuring, or they were measuring beta-lactamase activity from AMP-C. And if you don't induce it, there's a very small amount. And if you add cefoxitin, you can see there's massive induction in PA01. Okay, and again, that's because the cefoxitin is causing that peptidoglycan fragment to be produced, transported through AMP-G, works with AMP-R, turns on AMP-C. In an AMP-G deletion mutant without cefoxitin, again, there's a background level. And most importantly here, if you add cefoxitin, there's no induction. Why? Because the inducer molecule can't get to where it needs to go. So the hypothesis was correct. AMP-G could be a target for inhibiting AMP-C production. And before I go on, don't hesitate to interrupt me with any questions, okay? So what's the effect of this? In PAL1, the MIC of ampicillin, of course, another standard beta-lactam, is 250 micrograms per mil. It's basically ineffective. But if you delete AMP-G so that AMP-C is no longer induced, now the MIC is reduced to something that is reasonable. So if instead of deleting AMP-G, we could interfere with its function, in theory, we would restore the usefulness of ampicillin, to this strain. So that's kind of the genesis of the project. Okay, I basically just said that. Okay, so back in 2017, the collaborators between Scripps and UF published a paper on identifying small molecules that in fact could do what I just proposed, inhibit AMP-G and restore the usefulness of beta-lactams. So again, they published this a few years ago. Okay, so I'm gonna go through this maybe a little quickly, but to me, again, being somebody new to antibiotic discovery, development, research, this was a fascinating story here. So over at Scripps, and I visited them when I first started on the project. Really beautiful facility. This is done by robotics. They have 1,563 well plates. So they're they have tiny little wells, and they have cases and cases of these things stored, and robots can go in with pins and pick up some of the compounds from these and do them. So they said this is ultra high throughput, and they screen over almost 650,000 compounds from their diversity drug library in singleton, so there were no duplicates. And they did this in 2 weeks. I mean, I don't even know how you accumulate a library of 650,000 compounds, but they have it, they screened it, and they did this initial thing in 2 weeks. So what did they do? They had a Pseudomonas culture, and they had what was an ineffective level of cefoxitin. It was ineffective because AMP-C was degrading it. And so the level that was ineffective was 200 micrograms per mil, okay? Then they basically, so they looked for compounds that would then, by just measuring the OD of the culture, Pseudomonas plus 200 micrograms per mil of cefoxitin with the compounds added, which of the wells were reduced in their growth. And this, if there was reduced growth, it could mean that the compound inhibited AMP-C and therefore let the cefoxitin kill the bacteria, or the compounds could have killed or stopped the bacteria directly. So that's something we'll come back to. Okay, and I just said that. Okay, so this is a graphical representation of the data, and so down here, this is reading activity, and this is how much inhibition of growth there was. So the higher you are, the less growth. And so you can see up here the reference control is ciprofloxacin, which of course caused no growth. It killed the Pseudomonas. And then, down here, there's just DMSO solvent. That's what the compounds are in. And then they came up with this cutoff rate, which according to the pharmacy industry, 11.1% was their mark. And again, I don't necessarily understand the rationale behind that, but you can see there were a lot of hits up here between the low control and some of them approaching, the high control. Well, the hit cutoff is 3 times the standard deviation. Okay. All right. So they got these things. So this is, shown here. And I'm going to move my camera so because there's light in the way. So I don't know if it's showing me or not anymore. So they started off with 650,000 compounds. There's the criteria, and they ended up with 2,600 things that decreased. They did a triplicate repeat, and you can see that a lot of them didn't repeat, but they went ahead and they moved forward with these 2,600 compounds, right? So now they said, we're not interested in any. Yeah, go ahead. Question? Okay. So they said, let's find those that will inhibit Pseudomonas without the cefoxitin. Those would be interesting, but they're not what we're looking for, right? We wanted to inhibit AMP-C. So they did what's called a counter screen. They did the absorbance, OD600 with no ceph, and you can see that 864 compounds inhibited the growth even without the cefoxitin there. Probably not, super interesting in terms of this. They put them aside for further drug development later. Okay. Then they did something. There are these compounds called pan-assay interference compounds, or PAINs, and this is a kind of a virtual screen. Apparently, the chemists there, they know which of these compounds might or might not have issues. There I will confess ignorance. I just took it at face value. But when they removed the PAINs from these 2,600 compounds, there were 110 remaining. I guess being on the outside, I would say, well, why would they have these pan-assay interfering compounds in the library in the first place? I don't know. But anyway, now they're down to 110, right? Now, if you demand that the inhibitory concentration 50, IC50, is less than 10 micromolar, which in the pharmacy realm seems to be the magic number for usefulness. There was only one compound remaining, as you can see, over here. But they put that aside and they said, well, let's continue to look at some of these 110 anyway, right? And so again, what they had, the criteria were, they inhibited Pseudomonas in the presence of cefoxitin, didn't inhibit it in the absence of cefoxitin. So it seems that it was inhibiting, it was somehow inhibiting antibiotic resistance. Well, so to measure that directly, they created a Lux fusion with the AMP-C gene in trans. And then they induced it with cefoxitin, and then they added the compounds to that, and they basically said, how do these compounds affect the AMP-C Lux, right? And so we're looking for a decrease, and here's a graphical representation inserted elsewhere in the chromosome via a TN7 system. Here's the promoter for AMP-C, here's the Lux fusion, right? So here we are, and you're growing the bugs, and the green is the normal wild type, and you add the cefoxitin, and as I showed you before, massive induction of AMP-C, right? And again, confirming with the blue line, if you have an AMP-G mutant that can't transport the inducer, cefoxitin did not massively induce the AMP-C. So that just is showing that this AMP-C Lux assay actually works in a way that they had proposed. Okay, so here is some data using that assay. Okay, if you have the AMP-C Lux and you add cefoxitin, you get induction. These are just relative light units. And if you add DMSO, there's not a significant decrease. And now if you add CCCP, which is a proton motive force uncoupler, you see massive reduction in AMP-C Lux. And why is that? Well, that's because that AMP-G transporter is ATP dependent, and the proton motive force uncoupler will reduce the ATP in the cell. So there's no energy to transport the molecule, and therefore AMP-C lux will be not. That's a positive control that we like to use. Okay, so here's one of the compounds that I'm going to talk about in a little bit. So I'm getting ahead of myself here a little bit, but this is just showing that, and by the way, this compound wasn't discovered in the first round, as you will see. But anyway, the bottom line is if you add this compound at a relatively high level, okay, 100 micromolar, it did significantly reduce AMP-C lux. So it did show, this result here does show that compounds can be assayed using the AMP-C lux assay. Okay, so they used that assay and they screened the 110 compounds after the panes had been removed. And you can see that 8 of them did in fact induce AMP-C lux expression. And of those, none of them reduced none of them reduced the hold it, let me, did I just get ahead of myself there? Oh, no, I'm sorry, yeah, the key sort of absorbance. None of them reduced the absorbance of the culture. None of them killed the bacteria. But they did inhibit the AMP-C lux. Okay, so there were 8 compounds that survived all of this stuff that was put in that paper. And here are the 8 compounds. So they published these pictures, these structures, and notice one of them was polymyxin B, which we know is an antibiotic that is actually used against gram-negative bacteria. It's not very effective against Pseudomonas. Its outer membrane is somewhat resistant. Okay, I'm not going to talk about these. Why? What do these 8 compounds have in common, other than the fact they worked in this assay? They're not clinically useful. You know, the chemists look at, so we rediscovered polymyxin, and then there's all these other chemicals over here, and the pharmacology wizards at Scripps looked at those and said, none of these are useful. So it was okay to publish their names and their structures because they were dead ends. If they weren't dead ends and they hadn't filed a patent, then they would have basically forfeited the patent, by publishing that. So this whole screen here did show that the system could work. It did generate 8 lead compounds, but none of them were something that were ready for prime time. Okay, that was the background. Okay, then I came into the project about 3 years ago. Okay, so the wizards over at Scripps, they do something called structure activity relationship around, or SAR, I'll abbreviate it. And so they basically look at the chemicals and they look at their structures. They know, they kind of look at them and analyze them, and they say, oh, this looks like a group, maybe we should change this, maybe we should do that. And so they took 5 scaffolds from those primary hits, and then they subjected them to more medicinal chemistry, as they say. And of course, that was all done at Scripps. They ended up with 118 other compounds. We called them round 2 compounds. And they kept, doing their stuff. They were doing the AMP-C lux. They were doing the growth inhibition assays. And again, they're using their expertise in medicinal chemistry to figure out what does and might not work. Okay, and so they initially gave us 10 compounds in 3 different chemical series, but I'll just tell you that, make a long story short, we went through multiple rounds of going back and forth and testing them and arriving at some compounds. And this is like an example of one of the things. So they looked at them for inhibiting the growth, and they looked at them for AMP-C lux. And you can see they wanted some in this area that didn't inhibit the growth of Pseudomonas directly, but they did want them that inhibited AMP-C lux. And you can see here they ended up from this one particular screen with 41 compounds, and here are their numbers. These inhibited both. They might have usefulness, but not in this particular project. These 2 inhibited growth, but they didn't inhibit AMP-C, so not useful here. And these 5 over here didn't do anything. They ended up in the dustbin. Okay, so the model, of course, is that they're inhibiting AMP-C, and the model is that they're inhibiting the AMP-G transporter. They could be directly inhibiting AMP-C like clavulinate, or they could be doing something completely different. Okay, so how can we study this? Well, there is that inducer molecule but we don't have it. And if we did, how would we assay it? You know, we'd have to do like mass spec or something. So that would be difficult. So let's develop a somewhat, well, I don't want to say high throughput, but let's develop an assay that's easier. Let's develop a fluorescent assay using an inducer molecule that's got a fluorescent tag on it. There actually was one available that was developed in 2016, but it was very expensive, and it was made by these guys, right? So the wizards over at Scripps, they looked at this and they said, oh, here's a compound which might be useful. And I'm just going to abbreviate it. It's called 5,6-FAM. So they actually synthesized this, and I'll just tell you, that was something that amazed me in working with these guys. They're like, oh yeah, you know, we can do this 10-step synthesis with all this chemistry, and you know, most of us watching this probably don't have happy memories of taking organic chemistry as undergrads, but that's what these guys do for a living. And so they made this 5,6-FAM, and they made it much cheaper, and of course they didn't charge us for it, so it was free to us. Again, it was a neat part of it. Well, here's a problem. 5,6-FAM, there we go. 5,6-FAM is a big molecule, and it was too big to make it through the porins of the Pseudomonas, right, to get to the AMP-G to get inside of the cell. So we had to do spheroplast. And for the non-gram-negative people out there, a spheroplast is where you basically strip the gram-negative bacteria of their outer membrane. And now the fluorescent inducer is exposed directly to the AMP-G, and if it gets transported, then the spheroplasts, which do have an intact inner membrane, they will now become fluorescent, okay? And so we basically made spheroplast using a very standard method. Here's it's not phase contrast, it's just a plain micrograph of the rod-like Pseudomonas. You make the spheroplast, and now you can see all these little spheres, because when you degrade their peptidoglycan, there's nothing to make them rods, and they just turn into little kind of like micelles. So that's inner membranes. So I thought that was kind of neat, doing old school bacteriology. Okay, so does this assay work? Okay, so here's the bacteria. If you throw the compound in, they fluoresce. If you throw in the CCCP inhibitor, they don't fluoresce. And if you have the AMP-G mutant, they don't fluoresce. The spheroplasts don't fluoresce because they can't transport it. So the spheroplast 5,6-FAM assay works. Okay, and I'm just showing you here an example of using that. And I'll tell you, it's kind of a pain to make spheroplasts, because if you don't do it just right, they pop. And if they pop, of course, they're not useful. And if you don't do it just right in the other direction, you don't get spheroplasts, and the whole cells don't work. But once you make a batch of spheroplasts, you can save them and store them and use them. So this is just an example of using that spheroplast assay, in this case, to screen a broad panel of compounds from Scripps. So again, here's the fluorescence. You add the DMSO negative control, no effect. CCCP inhibits the transport. And then you add all these different compounds, and you can see some of them inhibited, some of them didn't, right? And these were all hits on AMP-G. On AMP-C. And here you can see I've highlighted compound number 4, which I'm going to call UFS, University of Florida Scripps number 4, did a really good job of inhibiting the transport of the fluorescent inducer. Number 16 was also pretty good. You're not going to hear about it anymore. It's very structurally similar to 4, and 4 was better. So we did look at 16 some. And these others, they just kind of got shelved. They weren't quite as good. So this is an example of using this transport assay to do some reasonably high throughput screening. And again, this is a little bit more direct function than looking at AMP-C, because that's the downstream. This is looking at transport of the inducer molecule. Okay, but I will note, we have not as of yet shown that some of these compounds, like compound 4, are in fact physically acting directly on the AMP-G transporter. We haven't done a BioCore and similar kind of biophysics assays that can detect whether these compounds are binding to that protein. And the fact that AMP-G is a transmembrane protein makes it difficult, to do that. So the model is that they're plugging up that channel, but we don't have any direct evidence, and that becomes important a little bit later on. Okay, so this is again looking at so we went through multiple rounds. UFS 4 was like round 3. They looked at it and then they said, oh well, let's do some substitutions here. They gave us like 5 more compounds. We looked at them in the AMP-G transport and the AMP-C Lux assay. They came up with a compound called UFS 18, and then we did this some more, and we ended up with another compound called 7G3. So these are our 3 lead compounds, which I'm going to focus on. So again, this is just showing those 3 compounds in the AMP-G transport assay. Again, I've already showed you that UFS 4 was reasonably good, UFS 18 was better, and 7G3 was even better. Okay, so inhibiting transport of that fluorescence probe. Now, this is a dose response of those 3 chemicals in the AMP-C Lux assay. So if you have induced AMP-C and you're just not inhibiting it with anything or even DMSO, you can see high activity. You throw in CCCP. Now note this is a log scale. CCCP inhibits AMP-C Lux. Why? Because it's inhibiting the transport of the inducer, because the AMP-G can't work because ATP goes down. Here was our first lead compound, UFS4, and kind of a shallow dose response. It worked better than CCCP. And of course, we can't use CCCP because it's toxic to us as well. And then this shows the next generation UFS18 was even better. Now at 100 micromolar, you can see, it's a decreasing activity by over a log. And look at 7G3 at 100 micromolar, it's decreasing it by almost 2 logs. But this is not physiological, right? The pharmacologists would shrug their shoulders. Even 50 is pretty high. So something down around this range is where the pharmacologist would like to be living, around 10 micromolar. Okay, but anyway, it does confirm that the compounds as they evolved did get better at inhibiting AMP-C. Why? Because they were inhibiting the transport of the small molecule, right? And so this is reading out gene expression using a Lux fusion. So what if you read beta-lactamase directly? And that's what we're doing here. This is a real pain because you have to do the extraction, protein determination, measure enzyme activity using fluorescent substrates for the beta-lactamase. Right, so here's the strain, and if you induce it with Ceph, you get 43 arbitrary units. Well, they're not arbitrary, it's micromolar per minutes per milligram. And if you would add CCCP, you cut it down by about 1/3. If you add UFS4, you cut it down some. If you add UFS 18 and 7G3, they cut down the enzyme activity. So that's why I'm saying some. This is not 0. It doesn't reduce it down to background. And this is going to be problematic, as you'll see. So these 3 compounds were doing what we asked them to do, but just not probably good enough. And you'll see that. So I'm going to skip a lot of negative results. Does inhibition of AMP-C, as I've here shown you with enzyme, translate into increased effectiveness of beta-lactams? So we did a bunch of synergy assays where we took the Pseudomonas and we gave them a sub-inhibitory concentration of a wide range of beta-lactams like methicillin, ampicillin, cefoxitin, and just a piperacillin, a whole bunch of them. And then we threw in these compounds in varying amounts, and we said, do they act synergistically and inhibit the Pseudomonas? Do they restore the usefulness of these beta-lactams? And the answer was no, they didn't. Very, very disappointing result. And so that reduction in AMP-C appeared not to be sufficient to allow the beta-lactams to now do their job. And so is there there seems to be a technical disconnection. Now let me just take a step back. So with these compounds here, we had to stop going back and forth doing more SARs, right? And so we arbitrarily stopped when we got to 7G3 because it seemed like it was doing a really good job. So as you can see here, what part of the solution is to go back with the Scripps guys and do some more rounds of SARs, because they can now look at the structures as they have evolved and try to come up with better compounds. But we had to take a pause so that we could write up and finish the studies on the compounds that we had. So again, it's not it's not a hard stop. It's a disappointing pause. And I guess this was like an eye-opening revelation to me, because my career up to this point in pathogenesis was, you can ask, how does this work? What's the effect of knocking out this virulence system that you've discovered in your favorite pathogen? How does it affect the host pathogen interaction? So that is like, I guess I'll call it knowledge discovery. And so basically, you're going to learn something. You'll learn that the system is really important in doing this or that at a cellular molecular level, or you'll learn that maybe it's not relevant under something, but you're going to learn something and you're going to get a result. But this is drug discovery. This is, can we come up with compounds that do something that we want, and can we make them eventually clinically useful, right? And sometimes you might go down a dead end. I think we can appreciate that something like 99% of drug discovery for antibiotics and bacteria, ends up in a dead end. I don't think this is necessarily a dead end, but the bottom line is, for all the work that I've been involved in for the past 3 years on this part of the project, has not yielded something that is even beginning to take to market. Okay, but again, as we move on, we can work with Scripps and have them, continue to come up with maybe better molecules. I'm going to shift gears here. Does anybody have any questions on that part of the story? Okay, so meanwhile, we're going to talk about type 3 secretion. So type 3 secretion is a way that some gram-negative bacteria inject proteins into the host cell. So here's the outer and inner membrane of your favorite gram-negative pathogen, Pseudomonas, Salmonella, many others, Shigella, E. coli. I mean, many of them have them. And they basically make a needle, and it attaches to the host cell membrane, and then they inject toxins or proteins into the host cells, and they do a wide variety of things. They can stimulate uptake, they can inhibit uptake, they can be cytotoxic, they can induce apoptosis, they can kill the cells, they can do a million things. Drives the med students crazy when I'm teaching them, because they want things to do one thing, right? I mean, one protein does one thing, but there's many different proteins. So one of the proteins that Pseudomonas injects is XOS. And there's a whole bunch of these exos. There's XOS, T, U, and Y, right? And they each have very specific things that are critical for virulence. So the type 3 secretion system of Pseudomonas is essential for cytotoxicity and virulence. It's been studied extensively. Shoguang Jin, my colleague, has actually published papers using Pseudomonas as a protein delivery system to inject proteins of interest into host cells that can change the differentiation of the cells. So interesting stuff. Okay, but why am I saying that? Well, that same AMP-R that is used to induce AMP-C also interacts with the regulatory system, again, widely found in gram-negative bacteria. In Pseudomonas, they call it RSM, but in most other bacteria, they call it CSR. And a colleague of mine down the road at the microbiology and cell science department basically discovered and has done immense work on the CSRA system. It basically regulates seemingly almost everything going on in these bacteria. But anyway, AMP-R works with RSMA to turn on the type 3 secretion system, which is highly regulated, right? That's why I said AMP-R is really a misnomer. It doesn't just regulate AMP-C, it does many things. So it really should have another name. So the hypothesis was, if that small molecule can't work with AMP-R to turn on RSMA, then the type 3 secretion system won't be turned on. And maybe some of these compounds have a side effect of inhibiting virulence. Okay, So let's look at some type 3 secretion. So this is a Western blot of that XOS type 3 secreted protein. What you need to know is that it's induced when you chelate divalent cations using EGTA. So this is PAK, which is a Pseudomonas strain, which is a hyper type 3 secretor. And we have the cell pellet down here. We have the culture supernatant. In the absence of induction, there's a little bit of XOS in the cell. There's none in the soup. You add EDTA, and it gets massively upregulated. You can see it in the cell pellet, but more importantly, it's exported into the supernatant. It's not being injected into anything. It's kind of just being injected into the supernatant. This EGTA tricks the Pseudomonas into thinking that they've contacted a host cell. Okay, so do our AMP-G transport inhibitors affect type 3 secretion in Pseudomonas? And the answer is yes. Let's just look at UFS4. These are just increasing amounts of UFS4 from 12.5 micromolar up to the huge dose of 100. And you can see that as you increase the dose of UFS4, the cellular XOS decreases reasonably, and it eventually disappears from the supernatant. Now remember that UFS18 and 7G3 were more potent than UFS4. And look at UFS18 at 12.5, it really is severely inhibiting export into the soup, and you get to 25 and higher, and there's none detectable. And look at 7G3, even at 12.5, there's none in the soup, and it's really suppressing the expression even in the cell pellet. So yes, these three compounds are in fact inhibiting type 3 secretion in Pseudomonas to varying degrees. So this opens up a whole new aspect of the project, which we've been spending a lot of time on, and that is, can they inhibit virulence? Now, we're not the first people to think about developing drugs that inhibit virulence as opposed to viability, as opposed to antibiotic resistance of bacteria. That's become a relatively hot topic alternative way of fighting infection, of inhibiting the virulence of the bacteria so that even if they're in us, they can't do us harm. And inhibiting type 3 would be one way of doing that for Pseudomonas. Okay, so XOS is a cytotoxin. So the question is, do these compounds inhibit the ability of Pseudomonas to be cytotoxic? And the way we do that is to take, these are HeLa cells, and we just basically took the monolayer of HeLa cells and stained them with crystal violet. And so you can see nice, thick, and then we solubilize them so we could read it in a spec. So here you can see the monolayer with no treatment, nice, and a lot of crystal violet staining the cell mass. You infect them with PAK, the cell monolayer basically was wiped out. If you try to protect them with DMSO, of course you can't, the cell monolayer is wiped out. But look, if you add 100 micromolar of compound 4, we protected the monolayer reasonably well. And if you add in compound 18, didn't work as well. There was a little bit of a disconnection here. This is just a graphical representation of measuring the OD, but I think this kind of shows it over here. So yes, to some degree, they could protect the monolayers. You know, when you give a seminar, you tend to want to present your best data. And I would say this one experiment here was the best that UFS4 worked. And again, this is a huge dose. A pharmacologist would never think about, you can't achieve this in an animal, right? So if you do a dose response and you get down around to 10 or so, it's not very pretty. So they moved in the right direction. They were inhibiting cytotoxicity, and 7G3, the third compound, did as well. So promising, but not ready for prime time. But the compounds came from a library, and as you can imagine, there again with the medicinal chemists, they know there are all kinds of side groups that and chemical properties that will enable small molecules to get into bacteria. be degraded, by endogenous enzymes. And so they're familiar with that. So they can look at these structures and go, oh, we need to get rid of that. We need to change this. Maybe if we add this, it might make them more stable, more highly transported or whatnot. So they're, that's going to be further rounds of SAR. So again, a tantalizing result. Okay, now type 3, as I said, is very widely examined. We weren't the first people to look at inhibition of type 3. One of my former favorite bacteria, as Chris said, Salmonella. I didn't study type 3, but actually the discovery of type 3 in Salmonella was done by my postdoctoral colleague when I was a postdoc in Roy Curtis's lab, Jorge Galan. Anyway, so this is just a Kumasi blue stain gel of supernatant of Salmonella. And these are just proteins that are in the soup. And you can induce the type 3 secretion system of Salmonella by changing their temperature from 25 to 37. So you can see here is the profile of 25. You change them to 37 in the presence of DMSO as a negative control. And here you can see the type 3 secreted proteins in Salmonella are CYPs. You can see these CYPs were induced. And if you throw in UFS4 and UFS18, they actually inhibited the CYPs of Salmonella quite nicely. What do these CYPs do? They help Salmonella invade epithelial cells. And so this is invasion of Salmonella into epithelial cells. And in the DMSO, marked as 100, there's the invasion. And you can see there are 3 lead compounds actually inhibited the invasion of Salmonella into the epithelial cells quite nicely. They almost seem to be working better in Salmonella than they were in Pseudomonas. I can say Roy Curtis is actually here at the University of Florida, and he still is doing Salmonella work. So we haven't yet given him these compounds to see if they interfere with Salmonella pathogenesis or not. That would be interesting because it would take me back about more than 20 years. Okay, some other virulence kind of associated phenotypes. One of them is biofilm. Okay, so here's a strain PAL1, and if you grow it statically, this particular case, and under these conditions, that strain doesn't make a good biofilm. But if you make a deletion of a gene called AMP-D, which is involved in that regulation, here's a really nice biofilm clinging to the side of the tube. If you throw in UFS4, the biofilm is gone. So UFS4 is capable of inhibiting the biofilm. If you throw in 7G3, which is related to UFS4 and shares many of its activities, it didn't inhibit biofilm. So now we're starting to get a disconnection between related compounds and their effects on the Pseudomonas that we don't understand. And it's beginning to raise issues in our minds that maybe they're not working exactly like we think they are. Another related thing is a compound called piocinin, which is green. PA14 is a hyperpiocinin producing strain. So here's the green color of the culture soup. You throw in UFS4, it inhibits piocinin. Again, 7G3 doesn't. These are kind of related phenotypes. And again, this is a tantalizing result that shows that these compounds are capable of interfering with not only AMP-C expression in Pseudomonas, type 3 secretion, yes, and other related kind of virulence-associated phenotypes, but now they're acting differently. So it's a very complicated story. Okay. Last kind of piece of data here. So a kind of animal model. I don't know if you've heard about it, Galeria melanella. That's the wax moth. And you can buy their larva from a company, which isn't too far from you all. It's North Carolina, Carolina Biologicals. I think it's in North Carolina. So way back in 2003, this group published a paper using the larva. They're not, I don't think they're technically caterpillars. I don't know. To look at Pseudomonas infection, because everybody was looking for alternative animal models, and they focused on type 3. I just want to point out one person in this group, Fred Ausubel. That should ring a bell, because remember back for this old people in the early days of molecular genetics, he, with colleagues from Harvard, published this tome. It was this huge book, And it was basically a lab manual for molecular manipulation. And I can't remember what the name of the book was, but everybody who was doing molecular genetic analysis, molecular manipulation had it. It was like the Bible of that. Anyway, so he got involved in this stuff here. Anyway, so what they showed in that paper was that type 3 secretion was essential for virulence in the model. And virulence basically consisted of killing the larva. And there's this noise coming through here. Okay, so what do? These are the larvae. They do look like caterpillars. And we basically inject them with like 10 to 20 microliters of a hypervirulent strain called PAK. And so we either grew the PAK with or without the compounds. So I will confess here, we're trying to throw things in our favor, right? And that is, we're going to inhibit the type 3 secretion system and whatever else with our compounds while the PAK is growing. And then we're going to mix it with the compounds and inject it into the larva at a high enough dose that it achieves a moderately high level of 50, oh, I'm sorry, that's supposed to be micromolar in the larva. Okay, and then we observe them in these little dishes for up to 48 hours. And we're looking to see, are they alive? And the easiest way of seeing are they alive is you tap on this and you see if they move. So I'm going to show you little snippets of videos. And you also look at their color, because when they die, they turn dark down here. So up here we have larvae that were just injected with PBS, no infection. Why? Because you do kill some of the larvae by injecting them. Let's see, this is supposed to be a little video. You can see, see, they're all, some of them are moving. And so the viability, as I'll show you graphically, was reasonably good. These guys aren't happy, as you can see, they're all dark. And none of them are moving. That's PAK, is the Pseudomonas infection, and they all died. And this is one of where we treated them with that UFS4 compound, and they're all dead. All right, so this is a graph showing percent survival. If you inject with PBS, you can see like one might have died. Our compounds didn't kill the worms, the larvae, which is good. We also showed that they didn't kill HeLa cells also. They weren't cytotoxic in themselves. You give them PAK and by 15 hours, they're dying, nearly 100% mortality. Could we protect them with UFS4? Yeah, some. Could we protect them with 7G3? No. There may have been a little bit of a delay, but the bottom line is the larva model didn't give us promising results, and we've done this a lot. So so here's where, so that's kind of where we are. So here are the problems. The compound, in terms of virulence, we had marginal biological effects in cell culture. That is, they protected the HeLa cells from cytotoxicity some, and they really didn't do a great job in the Galeria model. And so that's one, kind of like they didn't just quite make the grade in the antibiotic resistance arm. They haven't quite made the grade so far in the type 3 secretion virulence inhibition model. But there's yet one more problem. These compounds are classified as salicylidine acyl hydroxides, and they actually were examined back in 2009. Now this is a post facto discovery we didn't know this when we didn't pay attention to it when we first started working with them down the road. And so they were, in 2009, they were discovered as being anti-type 3. And they worked in Pseudomonas, Salmonella, Yersinia, Chlamydia, and other bacteria. And they even had activity against HIV in cell culture. And one of them was called INP341, which did inhibit virulence of Pseudomonas in mice, but it never made it to clinical trials. And that usually means something got disconnected between the mouse studies and going into human trials. So our conundrum here is, are our compounds better than this apparently failed compound and its brothers and sisters from the time, or are we in a river going over a waterfall? And we won't know that until we follow up on it in our work with work with scripts. So what are we planning on doing here? So I haven't left you with a really happy ending. It's a story. That's why it's the long and winding road. We're not to the end of the road, but we are planning on continuing on with scripts. In terms of the anti-virulence arm, we do have IACUC approval to look and see if our compounds can be protective in a mouse model. The hope here is that if we take mice that are more complicated than larvae and cell culture, they may be able to kind of fight the Pseudomonas, and the compounds inhibiting virulence to some degree might actually push them over the edge. I will confess, I'm not super optimistic about that. The Scripps people did look at 2 of our compounds in mice, and the pharmacology is not super promising because the compounds weren't selected for stability in animals, right? They would have to do some more chemical wizardry to make them work. And we will do further rounds of structure analysis as we move forward with them. Same story with the AMP-G, AMP-C. We need to look and see if we could come up with better compounds, look and see if they can be active with other drugs. And of course, a mechanism of action, we need to do some RNA-seq of treated Pseudomonas to see what branches of genetics and physiology, metabolism, et cetera, are being affected. And when I say epigenetic analysis, I mean, let's knock out genes that might be involved in certain pathways and see how that affects these. And the other thing, which I should have listed down here for mechanism of action, is some kind of biophysical analysis of the compounds directly with AMP-G to confirm that they are in fact directly acting on AMP-G as we had hypothesized, because we haven't shown that. Some of those results that I showed you, particularly with affecting pyocyanin and type 3 and biofilm formation, do imply that they may be doing some other things. So there's still a little bit of a black box there. So with that, I will end. And thank you for your attention. And I'll take any questions. Like I said, to me, the bottom line from being a molecular pathogenicist to kind of developing antibiotic resistance person is, when you're out with drug discovery, sometimes the thing that you learn is that the drugs that you have are not good enough, and it just may be that the hypothesis here, although it worked qualitatively, may not work quantitatively to actually have clinical usefulness. Okay, so I'm looking at a question from Chris. Can these compounds be encapsulated for improved delivery to decrease the dose? Well, don't forget, we want them to get into the Pseudomonas. So if the encapsulation helps them in the mice and or the larva, yeah, I don't have any idea what's going on in the larva. Are they being degraded? What's their half-life? The pharmacokinetics in the mice that Scripps did, it kind of said that if we gave a really whopping dose, that they got down to 10 micromolar within 24 hours. And you can see that in vitro, 10 micromolar wasn't doing really great things. So we would really be relying on the Scripps medicinal chemistry wizards to really soup up these lead compounds and see. And as I mentioned way from the beginning, there were a lot of compounds that had direct inhibitory activity against the Pseudomonas, and they were basically put on the shelf because they didn't fit what was being studied here. So that does represent, I guess I'll say, kind of a salvage, of the project. There may be some things there that are completely unrelated to this in their mechanism of action that could be clinically useful.
Let me ask you an immunological question. Cystic fibrosis patients do make high levels of antibodies to beta-lactamase, but they're not protective. Why do you think that is?
Wait, you mean?
Yeah, go ahead.
No. So you said that the cystic fibrosis patients.
They make antibodies to beta-lactamase.
Oh, they
But-
do. Well, okay, probably because the bacteria are making a lot of beta-lactamase, and it's present in the periplasm, and things in the periplasm can leak out of the cells pretty easily when the cells are dividing, or possibly getting killed by other antibiotics. So I wouldn't doubt that they'll make antibodies to beta-lactamase, but the antibodies will not be able to get to the beta-lactamase because it's in the periplasm, and the antibodies are way, way, way too big to get into that site. It's kind of like Pseudomonas is naturally resistant to bacitracin and vancomycin, which act at the peptidoglycan, because those are very large antibiotics and they're way too large to get through the porins of the Pseudomonas. So we can target the outside surface and secreted toxins with antibodies, but we can't target inside parts with antibodies. If they could be micro-injected into the periplasm and they could neutralize the beta-lactamase, that would work. But I don't think there's a reasonable way of doing that.
That's the problem. I think
Yeah,
patients make antibodies to virtually all Pseudomonas antigens,
yeah,
but they're not helpful.
yeah, yeah. Yeah, kind of along that line, I remember we had a College of Pharmacy symposium here, and there's a line of research where there are some large molecules that do get transported through the outer membrane, these siderophores that transport iron, and so they have complicated transport systems in the outer membrane that actually can transport large things. And so there's a guy, he used to be at Harvard, I think he's retired, or maybe he's now at a company, but he basically started linking siderophores to some antibiotics like bacitracin and others, and they actually did get transported through the outer membrane, and it seemed to give them a heightened activity. But that, I don't think that still wouldn't work for an antibody, which is way, way, way too big.
Thank you.
Sure.
Any other questions?
Yeah, I see that Wei Zhang,
Yeah, Wei.
he has said that they're trying to develop small molecules in microbiome studies.
Yeah.
And I guess I would say good luck because
Oh, no. That's a
well,
way.
I have to, I did pathogenesis for 30 years, and I must confess that I don't think a patient has benefited from my my studies. I didn't develop a drug or a diagnostic, and none of my, but I think some of my research did end up making it, possibly. I know it did make it into some textbooks, so I feel good about that. But most of us basic scientists, we're happy to contribute to the cloud of knowledge. And I know that people can take things that we discover and apply it to something completely unrelated. So I get a lot of satisfaction, out of that. So I have no conflicts of interest because there is no company involved. But if there was, at this point in time, I wouldn't be buying stock in the company anyway. So,
But my question is, do you think it's possible to find a small molecule inhibitor for some specific bacteria?
oh, I think so.
Oh, you
I think
think
so.
that's good enough, right? That's because antibiotics wouldn't work for long term. We need to develop small molecule inhibitor to prevent the bacteria like colonization or translocation, either way. It's very, very useful technology.
Yes, I agree. And if you can target a very specific pathogen that's causing trouble and leave the rest of the microbiota intact, that's really ideal. And there are a lot of targets, like you said, let's just say colonization. If we could inhibit the adhesins of uropathogenic E. coli, we could decrease urinary tract infections. And I know there's a lot of work going on because we understand that molecular interaction between those pili and the receptors on the uroepithelial cells. But as we've kind of discovered here, things that work in cell culture or work at a molecular level, it's just a whole quantum leap to go into animals where the compounds are stable enough and they get transported to where they need to go, and you can get them there in the high levels that you want without being toxic, because they all eventually do have some toxicity.
Okay.
So, you know, it really, this experience has really opened my eyes, you know, to how difficult it is in drug discovery. Yeah, I remember decades ago, I was reading a series of papers from Abbott Labs up in Chicago. And, you know, when I teach med students about and grad students about antibiotics, why don't we target lipopolysaccharide? I mean, polymyxin B kind of does, but why don't we target LPS biosynthesis? That would be a great target. And they came up with drugs that inhibited LPS, and they killed gram-negative bacteria. But when they made the jump to animals, they completely fell apart because they just didn't do the job in the animals. And apparently the chemists weren't able to work with that, because that was decades ago and nothing has come of it in that time. So they're just at least 99 dead ends for every one that ends up working.
Okay, thank you.
Sure. But that's a good, you know, sniper rifles to pick off the pathogen of interest is much better than shotguns, which is what we generally use, that kill everything and then cause diarrhea and you know, yeast infections and other things.
Yes, great. Thank you.
Thanks. I think we should wrap it up. We're like 9 minutes over, but thank you, Paul, for
Okay, my
talking
pleasure.
today, and I will see you around.
Yes, yes. Okay.
Okay.
Well, everybody enjoy the long holiday weekend.
Okay.
Thank you.
It's been a pleasure.